This PowerPoint will give you an introduction to what is called the central dogma of biology or central dogma of molecular biology. It's really an explanation of how our cells use the information that's stored in DNA to make an actual product. So I want to give you a little bit of background information about the molecules that we're dealing with and a bit of a roadmap. So DNA is, it's a nucleic acid. It's a molecule. It's a really long molecule. And within our cells, each of our cells has 46 DNA molecules. And we call each of those molecules a chromosome. So the specific regions of DNA on that molecule we call genes. And each gene codes for a very specific product in the cell. And really all genes code for proteins. So proteins make us who we are. They make us human. They make us individuals. And so that really is the purpose of DNA, is to code for proteins. So some examples of different types of proteins. Some are structural, like collagen. Some are catalytic, like all enzymes. Some are transport, like hemoglobin, which moves oxygen around through our bodies. Some help with defense in our immune system, like antibodies. Some help with communication from one part of the body to another. So hormones do that. Some help with movement, like actin or myosin. And humans have between 20,000 and 25,000 genes. So if you remember, we have 46 chromosomes, and they actually come in pairs. And so we only have 23 different kinds of chromosomes. So on those 23 different kinds of chromosomes, we have to have 20 to 25,000 genes. So, some chromosomes have over 1,000 genes on that actual single DNA molecule. So like I said, you have 2 copies of each gene. We'll get more into this later in the unit, but one of those copies came from your mom and one came from your dad. Now, sometimes the 2 copies are identical. So for some proteins, maybe collagen, actin, humans kind of all code for that the same way and make the same exact protein. But for other things, for example, a gene that maybe codes for your hair color, your mom and your dad might have different versions of those genes. And we call those different versions of the same gene alleles. How a cell actually uses genetic information, so how it uses that code, is called gene expression. So I like to use an analogy to explain this process. Let's say that you are at home, this is before COVID-19, but you're at home and you realize you have all the ingredients to make a pie. But you only have enough ingredients to make a single pie, and you've looked around, you can only find a quarter. So if you mess up, you don't have enough money to go grab any more ingredients. You don't know how to make a pie, and your phones aren't working, your internet's down, you've looked through all the cookbooks in your house and you can't find a good pie recipe. So what you do, you grab your quarter, put it in your pocket, you get on your bike, and you ride over to your library. It's still open. So in the library, we have basically all the information for anything we could ever need. So you go to the cookbook section and you look around and you find the perfect recipe. It's in this giant, old cookbook that you're not even allowed to check out of the library because it's so old. But you find a single recipe that you really want. So you can't take that book out of the library, but remember you have that quarter. So you make a photocopy of that recipe. And so now you just have that single piece of paper that has the recipe on it. So that's really easy. You can fold it up, you can stick it in your pocket, and take that home with you. It's no big deal, it's not heavy. So you ride your bike home, and then you get out all your ingredients, you read the recipe, and you make that pie. So what does this have to do with DNA and with our cells? Well, the library represents all the DNA in our nucleus. So all those books have all that different information that we could possibly want, but those books never leave the library. The process of photocopying just a single recipe or a single item of information is called transcription. So it's similar to our cells just making a copy of the single gene that we want. And that copy, that photocopy, is the same as RNA, and specifically mRNA in our cells, and that can leave the nucleus. Then when our cells actually read that recipe or read that mRNA, that process is called translation. And that message or those instructions in this weird code is translated into another form, and it's basically a chain of amino acids which make up the protein, which is the product. Now there's another organelle that's helpful in this process, and that is the ribosome. So the cook in this analogy is the ribosome. Now if we take a look at this image here on the left, that's showing a molecule of RNA. On the right is DNA. So you're familiar with the structure of DNA already. RNA is similar, but there are some major differences. So one of those major differences that probably pops out at you right away is that DNA is made up of two strands, and they're held together in the middle with those hydrogen bonds between the bases. RNA is just a single strand, so it still has a sugar backbone or sugar-based backbone, and it has nucleotides sticking out towards the center, but they don't have any of those complementary nucleotides, bases on the other side, so there's not a double strand. Also, if you look at the 4 bases that are in DNA, we see C, G, A, and T on the right there. On the left, we also have C, G, A, but then instead of thymine with the T, we have uracil represented by a U. So that's another major difference. The third major difference is when we look down here at the names DNA and RNA, the D in DNA stands for deoxyribose. That is the sugar that is in that sugar phosphate backbone. In RNA, the sugar that makes up that sugar phosphate backbone is ribose. So the R stands for ribonucleic acid.